Figure 4.5, Cell Structure. Figure 4.5, Cell Structure. Cells are defined from the exterior environment by enclosing themselves within a plasma membrane. Plasma membranes are a combination of phospholipids and proteins. The phospholipids act as the barrier, while the proteins give function to the membrane. The nucleus is usually the largest organelle of the cell. It acts as the command center for all cellular activities. It's surrounded by a nuclear envelope, which is a double layer of phospholipid membranes. When the nucleus is stained, it's often possible to see the nucleolus, a region that's involved in the manufacture of ribosomes. Surrounding the nucleolus is chromatin, a mixture of protein and DNA. Except during specific times during the cell cycle, the DNA is not usually visible, but it is organized into structures called chromosomes. Attached to the nuclear envelope is a system of membranes collectively called the endoplasmic reticulum, or ER. The endoplasmic reticulum comes in two forms, one with ribosomes attached and the other without. The ER with ribosomes attached is called the rough endoplasmic reticulum. Its primary purpose is the manufacture of proteins associated with membranes, such as the proteins in the plasma membrane. The other form of ER is called smooth endoplasmic reticulum. It mainly produces lipids. The endoplasmic reticulum is a major component of the endomembrane system in the cell. Ribosomes are the protein synthesis machinery of a cell. It's here that the cell manufactures all of the proteins needed for cellular function. Sometimes many ribosomes may be involved in making the same protein simultaneously. This is called a polyribosome. All cellular activities require energy. The mitochondria are the powerhouses of the cell. It's here that sugars and other organic molecules are oxidized to form the majority of ATP needed by the cell. These processes involve oxygen and produce carbon dioxide as a waste product. Another important component of the endomembrane system is the Golgi apparatus. Think of the Golgi as a processing and shipping center. Molecules that are being exported from the cell pass through this organelle for final modifications. The Golgi also makes some of the other organelles of the endomembrane system. Materials typically come to the Golgi packaged in general use vesicles. Like trucks at a distribution center, there's a constant movement of vesicles to and from the Golgi apparatus. Two of the specialized vesicles that are produced by the Golgi apparatus are peroxisomes and lysosomes. Peroxisomes are involved in some detoxification activities within the cell, and also in the breakdown of fatty acid chains. Lysosomes are digestive and recycling centers. It's here that the cell digests incoming food into monomers. Lysosomes can also be used to recycle worn-out or damaged organelles within the cell. Both peroxisomes and lysosomes are considered parts of the endomembrane system. Another important component of the cell is the cytoskeleton. This is a dynamic structure that responds to the needs of the cell. Components of the cytoskeleton are involved in maintaining the shape of the cell, moving materials within the cell movement, and in cell division. There are three classes of molecules in the cytoskeleton. They are microtubules, actin filaments, and intermediate filaments. The role of centrosomes and centrioles is still a mystery to scientists. These structures are associated with the cytoskeleton of the cell, specifically the microtubules. They're known to be involved in cell division, but not all eukaryotic cells have these structures. Eukaryotic cells share many characteristics, but there are some important differences. Figure 4.5b illustrates a typical plant cell. Can you spot the similarities to the animal cell? These organelles are found in both plant and animal cells. However, there are a few important differences. Plants are autotrophic organisms that make their own food to survive. The organelle where this occurs is the chloroplast. Chloroplasts of plant cells are green due to the presence of a pigment called chlorophyll. A typical plant cell may have several dozen chloroplasts. Plant cells also have a cell wall, located just outside the plasma membrane of the cell. The cell wall is a rigid structure made of polysaccharides, and it serves to provide structure and protection for plant cells. Cell walls are also found in many other types of eukaryotic cells, but not animal cells. One of the more conspicuous differences between the plant and animal cell is the large structure in the center of the cell. This is called the central vacuole. It varies in size and shape depending on the type of plant cell. The central vacuole is a storage site for many cellular products. It can also be used to regulate the fluid levels of the cell so that the plasma membrane is kept tight against the cell wall. The examples in these diagrams are provided to give you a general idea of the structure of eukaryotic cells. Variations on these designs are common in plants, animals, fungi, and protists. These variations allow the cell to adapt to specific environmental requirements, or if the cell is part of a multicellular organism, specific requirements of the tissue where the cell resides.